In 1953, as Sir Edmund Hillary stood atop Mount Everest, two scientists, James Watson and Francis Crick, were on the verge of a discovery that would rock the very core of our understanding of life. They revealed the double-helix structure of DNA, a breakthrough that not only revolutionized biology, but also challenged the prevailing theory of evolution. For years, Darwinian evolution taught us that life evolved through random mutations and natural selection. However, as scientists delve into the intricacies of DNA, they are discovering something astonishing. The genetic code is far more complex than anything humans have ever created. Each human cell contains approximately 3 billion genetic letters, equivalent to 12 complete sets of the Encyclopedia Britannica. This complexity begs the question, can such intricacy arise from random processes, or does it hint at an intelligent design? Decoding the human genome has revealed that DNA operates as a sophisticated language, complete with grammar, semantics, and purpose. Bill Gates famously compared DNA to a software program, except far more complex than anything humans have devised. This level of complexity poses a significant challenge to the evolutionary model. How could such a precise and intricate system develop solely by chance? The average error rate in DNA replication is one mistake. But 10 billion letters, a feat no human typist could achieve. Furthermore, no evidence suggests that one functional information system within a cell can gradually evolve into another. The notion that life's complexity could emerge from random mutations is increasingly seen as implausible. Even prominent scientists, like the agnostic Francis Crick, have admitted that the origin of life seems nearly miraculous, given the countless conditions that must align for it to occur. These findings have profound implications. More scientists are now questioning the sufficiency of Darwinian evolution as they grapple with the complexities of DNA. The evidence suggests an intelligent source behind the genetic code. This shift in perspective is not merely an academic debate. It challenges the fundamental worldview that has dominated science for over a century. As we stand on the precipice of a new understanding of life, it becomes clear that the discoveries within the cell are not just scientific milestones, but also a call to reconsider the origins of life itself. The intricate design of DNA implies that we may not be the result of random chance, but rather the product of purposeful creation. This realization beckons us to explore the possibility that intelligence, not randomness, is the true architect of life. Genetic recombination, a fascinating process that occurs during the formation of sperm and egg cells, aligns with biblical accounts of creation by emphasizing initial diversity within humanity. According to the biblical narrative, all humans trace their lineage back to Adam and Eve, the original couple created by God. In this context, genetic recombination acts as a mechanism that introduces variation in the offspring, in harmony with the idea of diversity established by a creator. This process, along with the occurrence of mutations, contributes to the incredible genetic diversity within the human race. Some argue that this diversity supports a biblical model of human origins, as it accounts for the observed genetic variation among modern humans. There are intriguing parallels between DNA and the biblical concept of creation that further reinforce this connection. One such parallel is the complexity and design of DNA. The intricate structure and the remarkable ability to store vast amounts of information within DNA are often likened to a sophisticated language, hinting at an intelligent design behind life. This aligns with the biblical view of a purposeful creator who intricately designed every aspect of life. Additionally, the concept of the creation of life itself resonates with the biblical narrative. In the book of Genesis, it is described how God created each creature to reproduce after its own kind. Similarly, DNA plays a pivotal role in ensuring that offspring resemble their parents through the inheritance of genetic traits. This echoes the biblical notion of intentional creation and continuity within species. The idea of divine craftsmanship is beautifully metaphorized in Psalm 139, 13-14, which describes God's creation of humans as a process of intricate knitting. This imagery mirrors the complex weaving of DNA strands that occurs during human development. It emphasizes the profound intricacy and care with which each human life is created, aligning with the biblical account of God's intentional craftsmanship. By exploring the parallels between genetic recombination, DNA, and the biblical concept of creation, we find remarkable harmony between science and faith. The complexity and design of DNA, its role in the creation of life, and the craftsmanship involved in human development all support the idea of an intelligent and purposeful creator. Genetic recombination serves as a fascinating mechanism that aligns with the biblical narrative of initial diversity within humanity. It contributes to the awe-inspiring genetic variation observed among modern humans, further reinforcing the biblical model of human origins.